When you picture a caterpillar turning into a butterfly, it is incredibly easy to just imagine it, like growing wings.
Right, yeah, like it just climbs up a branch, zips itself into a tiny, cozy sleeping bag, and, wakes up ready to fly.
Exactly. We naturally gravitate toward that image because we like biological processes to be neat, I guess. You start with a base model, bolt on some upgrades, and you're good to go.
The classic children's book version of Metamorphosis.
Yeah.
But the biological reality is actually a lot more like a sci-fi horror movie.
Oh, completely. Because when that caterpillar enters its chrysalis, it doesn't just sprout wings. It undergoes this brutal process called hystolysis. The insect literally dissolves.
Yeah, its muscles, its digestive organs, its brain, it all just melts down into this highly fluid cellular soup.
It's the ultimate developmental reset button. Everything that made up that terrestrial leaf-chewing caterpillar is liquefied to provide the raw materials for the adult butterfly.
They're practically two entirely different organisms, sharing a single life cycle.
Which brings up a massive, glaring paradox that we are unpacking today. If the brain is completely liquidated into this organic soup, how could any memory possibly survive that level of total physical destruction?
Right, it seems impossible.
I mean, think about it for a second. It's like demolishing a house all the way down to the dirt foundation, pouring a completely new concrete building, and then expecting the old smart home settings to still be programmed into the walls. You would expect a totally blank slate.
And for a very long time, the scientific paradigm assumed exactly that. Complete erasure of the larval experience. Because, you can't maintain a hard drive if you melt a computer.
But the crazy thing is, and this is the big twist, the chrysalis is not a blank slate at all. Not only do memories survive this pupal reset, but new research shows butterflies might actually pass those learned fears down to their offspring.
And even their grandchildren, which is mind-blowing.
Yeah. And they do it without changing a single letter of their underlying DNA.
It completely upends how we think about biology and memory. And honestly, the researchers behind this breakthrough are just as surprising as the findings themselves.
Oh, for sure. We are talking about Joe Nagai, who at the time this began was an 8-year-old student from Kobe, Japan, and Dr. Martha Weiss, a renowned entomologist at Georgetown University. Welcome to Deep Cuts and Deep Thinking.
Thrilled to be here to dig into this one.
We are going to explore how a child's backyard curiosity challenged a long-standing biological paradigm, how this unlikely duo teamed up across the globe, and, what their discoveries mean for the very nature of memory.
It is just a phenomenal story about the scientific method in its purest form.
So to understand how this all started, we have to look at a kitchen counter in Kobe, Japan. Joe Nagai was raising Asian swallowtail butterflies. The scientific name is Papilioxeuthis, right, in his own backyard.
Just, feeding them, watching them grow, standard nature kid stuff.
A completely normal hobby. But Nagai noticed something highly unusual. When he released the adult butterflies that he had hand-raised from caterpillars, they didn't just fly away into the trees.
They lingered.
Yeah, they hung around him, and they even returned to land on his hands.
Which, from an ethological perspective, is a really profound observation for a kid to make. It sparked this hypothesis in his mind like, did these butterflies remember me?
Did they retain a memory of the giant hands that fed them when they were tiny caterpillars, even after, turning into soup?
Most kids would just assume they had some sort of Disney princess connection and move on.
Exactly. But Nagai went straight to the scientific literature. He dug up a landmark 2008 paper written by Dr. Martha Weiss, which had proved that tobacco hornworm moths could actually remember things from their caterpillar stage.
So, in the spring of 2022, this 8-year-old second grader sits down and writes her a letter.
A detailed 4-page handwritten letter, packed with earnest scientific curiosity. Dr. Weiss later mentioned it ended with 2 bold exclamation marks, just radiating absolute enthusiasm.
And asking to collaborate. I mean, Dr. Weiss is a veteran scientist at a major university, getting a handwritten letter from a child on the other side of the planet.
She was incredibly supportive, but she definitely had some serious academic skepticism at first.
Oh, of course. Replicating complex laboratory conditioning is notoriously difficult in a controlled university setting, let alone in a family kitchen. So she initially suggested a much simpler experiment.
Like a backup plan?
Yeah, she advised Nagai to try training the adult butterflies to associate specific colors with a sugar water reward. A nice, safe, standard science fair project.
The kind of thing that won't end in tears. But Nagai politely but very firmly declined. He totally refused to give up on his caterpillar memory hypothesis.
He essentially replied that he really wanted to prove his swallowtails could remember what they learned as caterpillars, and he pleaded for her help to design the experiment.
Okay, let's unpack this, because this is the moment the story shifts from a cute backyard observation into genuinely wild, unhinged experimental design. He is doing MacGyver-level science at his kitchen table.
It really is incredible. In Dr. Weiss's original university lab study, they used toxic chemicals like ethyl acetate and high-voltage lab shockers to train the caterpillars.
And Nagai couldn't exactly order industrial toxins to his house.
No, that would certainly fail the parental safety check. He had to innovate with household items, so for the scent, the conditioned stimulus, he replaced the toxic chemicals with natural lavender oil.
Which is highly aromatic, biologically benign, and crucially, naive swallowtail caterpillars have absolutely no natural fear of it, right?
Exactly. But he still needed a way to create a negative association. A mild aversive stimulus.
So instead of a high-voltage lab shocker, he repurposed a personal nerve stimulation device. You know, the kind of mild electronic pulse generator that people buy at the pharmacy to relieve lower back or muscle pain.
Brilliant problem solving on a budget. But those consumer devices are specifically designed for human skin. They require a closed electrical circuit across conductive, salty tissue to work.
Right, you can't just tape the sticky pads to a plastic table, put a tiny caterpillar on it, and expect a current to flow.
No, it wouldn't work at all.
So he figures that out and creates what I am calling the arm interface. Nagai takes the sticky electrode pad from this mild electronic pulse generator and slaps it directly onto his own bare arm.
Wow.
Yeah. And then he physically places the caterpillar underneath the pad, resting right against his own skin.
So by doing that, when the device activates, the electrical current flows through Nagai's arm and the caterpillar's body simultaneously.
The circuit is closed by the moisture in his skin. He is literally zapping herself alongside his test subjects.
The sheer dedication of this kid. He did this so he could feel the exact physical intensity of the pulse and dial in the settings perfectly.
Yeah, he cranked the device up to level 4. And the way he knew level 4 was the magic number is so fascinating.
He was observing the caterpillar's anatomy for a specific biological response. At level 4, the caterpillars consistently popped out their osmeteria.
Those little fleshy, orange, defensive stink glands that come out from behind their head when they feel threatened?
Right, it's their primary defense mechanism. Triggering the osmeteria meant Nagai had found the exact threshold where the caterpillar was intensely startled, but totally safe and uninjured.
It was a highly salient stressor. And to give you an idea of how that feels, a reporter named Annie Rosenthal later tried the same device on her own arm while covering the story.
And what'd she say?
On just level 3, she reported feeling a huge shudder that made her hand physically jump involuntarily.
And Nagai is taking level 4 hits to his arm over and over just to ensure a rigorous classical conditioning setup in his kitchen.
It's wild. So he trains the caterpillars, they smell the lavender, they feel the mild pulse, they learn to hate the lavender. Then he lets them pupate. They turn into that cellular soup and emerge as adult swallowtail butterflies.
Which brings us to the million-dollar question. Did the memory survive the meltdown?
To answer that, Nagai utilized a Y-maze choice tube. It's essentially a piece of testing apparatus shaped like a capital Y.
Right, the adult butterfly is placed at the bottom stem. At the top of the two divergent arms, there are sugar water feeders to encourage the insect to walk forward.
And one arm of the Y is completely scent-free, while the other arm contains a cotton pad infused with that natural lavender oil.
When Nagai tested the control butterflies, the ones that were raised normally and never trained, they split evenly. 50% went to the unscented arm, 50% went to the lavender arm.
A perfect coin toss. It proves they have no natural preference or aversion to the smell.
But the conditioned butterflies, the ones that learned to fear lavender's caterpillars while getting zapped on Nagai's arm, showed a massive aversion.
Yeah, 70 to 80% of the conditioned cohort entirely avoided the lavender-scented arm.
Which is a statistically massive result. The memory actually survived hystolysis.
Which brings us right back to our central paradox. How? If the early-born cells turn to mush, there has to be a physical survivor, right? Are there cells that just, skip the hystolysis phase?
What's fascinating here is the neuroanatomy of the insect brain, which holds exactly the loophole you are describing. We have to look at a structure called the mushroom bodies.
The mushroom bodies?
Yeah, these are the paired processing centers in the insect brain that handle olfactory memory, how smells are processed and remembered. The core of these mushroom bodies is made up of thousands of individual neurons called canyon cells.
And these canyon cells are categorized by their developmental timeline, right?
Exactly. They are divided into early-born canyon cells and late-born canyon cells. The early-born neurons are formed when the caterpillar is very young.
And during pupation, those early-stage neurons absolutely do get pruned. They retract, synapses break, and they are histolized.
Any memory stored solely in those early pathways is completely erased.
But the late-born canyon cells, those are generated in the final mature larval stage, just before the caterpillar forms the chrysalis.
And because they are still actively maturing when the pupal meltdown begins, they are biologically exempt from the pruning process.
So they don't turn to soup.
No, they persist completely intact through metamorphosis. They essentially act as a physical structural bridge, keeping that specific neural circuit alive into adulthood.
Going back to our earlier analogy, it really is like replacing every single piece of hardware in a computer. The motherboard, the fan, the power supply, except for the exact hard drive where one specific file is saved.
The brain rebuilds itself around that surviving hard drive. That structural persistence is what allows the adult butterfly to recall a danger it only experienced in a completely different physical body.
Documenting that biological bridge is an incredible scientific achievement for anyone, let alone a kid at his kitchen table. But the story goes one massive step further. We have to talk about inheritance.
Yes, because surviving the chrysalis is one thing. Passing it on to the next generation without changing the DNA is something else entirely.
So Nagai decided to breed these conditioned butterflies. He takes the adults that were trained to fear lavender, and he lets them mate.
They produce offspring, which biologists call the F1 generation, and then those offspring produce grandchildren, the F2 generation.
And the environments these new generations were raised in are vital to the whole experiment. The F1 and F2 generations were kept completely naive.
Right, they were raised in a totally normal, peaceful environment. They were never exposed to the mild electronic pulse generator, and they never smelled lavender oil while they were caterpillars.
They had zero personal experience with the stressor. Yet, when Nagai put those completely untrained descendant butterflies into the Y-maze, they still showed that exact same 70 to 80% avoidance of the lavender scent.
They avoided it at the exact same rate as their conditioned ancestors.
If they weren't trained, and their underlying DNA code didn't mutate, where's that memory coming from?
This brings us to the frontier of non-genetic memory, specifically transgenerational epigenetic inheritance.
It's just bizarre to think about.
To understand how bizarre this is, think of how biologists used to view the body. They believed there was a strict firewall between your somatic cells, your body's daily experiences, and your germline tissues, your eggs or sperm.
Like if you get a scar on your arm, your child isn't born with a scar.
Exactly. Your daily experiences were thought to be completely walled off from reproduction. But this research implies there is a back door in that firewall.
So how does the information get through?
The current understanding is that during the traumatic conditioning, the extreme bodily stress generates specific chemical molecules. These parental stress molecules manage to sneak through that firewall.
And they go where?
They are transported and deposited directly into the gametes. So when those gametes form the next generation, those chemical tags alter the offspring's gene expression.
They don't change the genetic code itself, but they change how the code is read.
Yes, essentially pre-wiring the descendant sensory pathways to be highly sensitive to that specific environmental hazard.
Here's where it gets really interesting, because this isn't just an insect quirk. If we connect this to the bigger picture, we see striking parallels in mammalian studies too.
Oh, absolutely. The most famous example is a fear conditioning experiment in mice, conducted by researchers Weiss and Cellular.
What did they do?
They trained male mice to associate a specific fruity smell acetophenone with a mild foot shock. And much like the butterflies, the offspring and the grand mice of those trained males were born with a heightened innate sensitivity to that exact same fruity smell.
Their brains literally had more structural space dedicated to detecting it, despite never being shocked themselves. The trauma shaped the biology of the descendants to protect them.
It's an incredible survival mechanism.
But if you're listening to this and thinking, wait, couldn't the lavender scent just have wafted across Nagai's kitchen and accidentally trained the baby caterpillars while they were growing? Well, you're thinking exactly like a peer reviewer.
This raises an important question about scientific rigor. Claims of inherited memories face incredibly high skepticism in the biology community.
Why is that?
There is a well-known reproducibility crisis in the field of epigenetics.
What does a reproducibility crisis actually mean for a scientist trying to publish?
It is a career-defining hurdle. It means that when other scientists try to run the exact same experiments, they frequently cannot get the same results. Critics point to issues with statistical power, arguing that the sample sizes in many epigenetic studies are just too small to be conclusive.
And there's publication bias too, right?
Huge publication bias. Journals only want to publish flashy, successful experiments, while the failed replications are quietly hidden away in desk drawers. With some of these transgenerational studies, statisticians look at the data and argue that the inheritance rates look, frankly, too good to be true.
And a kitchen table with open windows and family members walking around is a variable nightmare. Ambient drafts, temperature changes, cross-contamination.
Exactly. If a single molecule of lavender reached those naive F1 or F2 caterpillars, the inheritance data is compromised.
So to get this published in a formal peer-reviewed journal, which Dr. Weiss and Nagai are currently working toward, they have to transition from kitchen table science to absolute laboratory lockdown. They are completely upgrading the methodology.
First, they need automated video tracking for the Y maze.
Because human observation is naturally biased.
Yeah, if you want an insect to turn left, you might subconsciously count a slight twitch as a left turn. A computer algorithm needs to objectively record which way the butterfly walks, eliminating the human element entirely.
And second, they need sealed environmental flow chambers, so they have to ensure absolute isolation of the sense with zero chance of drafts or leaks.
And finally, they need genetic mapping. Behavioral observation is just not enough to satisfy the skeptics anymore.
They want the receipts.
They do. They have to map the germline tissue of the swallowtail to prove exactly where and how those stress molecules shifted the gene expression. They need to show the molecular receipts.
It is a massive undertaking. Just the sheer fact that we are even discussing the molecular verification of a second grader's hypothesis is incredible, and the scientific community has absolutely taken notice of this duo.
The reception has been global and overwhelmingly positive.
Joe Nagai and Dr. Weiss presented this research together at the 2024 International Congress of Entomology in Kyoto, Japan. Think about that visual. A young student standing on a global stage alongside a veteran scientist.
They even briefed the Japanese crown prince on their findings.
The story blew up. It hit major media outlets, including a huge feature on WNYC's Radio Lab.
Going from a handwritten letter to briefing royalty is the ultimate scientific journey.
So what does this all mean? When you step back and look at the entire scope of this project, this is a celebration of curiosity-driven science.
It really is. It proves that you don't always need a multi-million-dollar grant to ask a profound question about the natural world.
A young student and a veteran scientist work together across generations and across the globe to show that metamorphosis is not a blank slate. It is a complex structural bridge.
It forces us to view life cycles and inherited traits through a completely different lens.
I want to leave you with a final thought to mull over as you go about your day. If a simple insect can pass down the physical memory of a single stressful event across multiple generations just to protect its young, well, what hidden survival memories might be echoing in our own human habits?
What modern anxieties or instincts are we carrying that aren't truly ours, but rather the chemical echoes of our ancestors' survival?
It's a profound question to walk away with. If you found this podcast interesting, please like and subscribe. Thank you for joining us today.